I started out being very interested in finding new therapeutic targets in the rhabdomyosarcoma. And we started with a sort of new idea, which was a lot of chemotherapy that we give really focuses on DNA, focuses on damaging DNA, creating mutations, and relying on cells to recognize that something has gone wrong in order to get rid of them. Instead of doing that, we decided to focus on proteins themselves. And there's a very interesting amount of literature that's come out in the last couple of years around these proteins called chaperones. And we think that cancer cells actually really depend on these chaperones to get their proteins made properly and to the right place. That was an idea that we wanted to test using drugs, and so we used an inhibitor of a particular chaperone called HSP70, and we actually found that by inhibiting the way that that chaperone binds to proteins, rhabdomyosarcoma cells very specifically seem to just give up and die. My goal is to try and get this to patients as quickly as possible, and that's been something sort of fun to think about. Part of me that's a scientist really wants to delve more into the biochemistry, but the part of me that's trained as a clinician really wants to try and get this into prime time as quickly as possible. It's really kind of opened new doors for me clinically, which I wasn't really expecting. I knew that it was going to get me great support to stay in lab and protect me from that, the sort of brain drain that happens when you sort of need to be constantly running back and forth. But it's actually allowed me to develop a clinical expertise, which was an unexpected side effect and has really been a lot of fun. And I think it's challenging to know how to be a great clinician who is also contributing to the field scientifically, because those can sometimes be at odds with each other. But to have had a project that was fully funded from the get-go, that really let me take a very basic science question and ask how we can start applying it to patients, has kind of let me run with those two groups of people. I can't really stress enough the idea that misfolding proteins can somehow turn into a cancer drug for kids with muscle sarcoma is crazy. And it's not something that I would have come to without the support of a lot of this basic science community that's out there. So to be able to work with a funding agency that's really trying to bring all of these people together, the opportunities you get from that are fantastic. I think that really sets it apart.